Hello everyone, I'm Zhiqian Zhai from UCLA. This is a joint work with Professor Yu Lei from the University of Michigan and Professor Yuying Xie and Rong Wang from Michigan State University. So we proposed a visualization method called Supervised Capacity Preserving Mapping, which is a clustering-guided visualization method for single-cell RNA sequence data. Our method is motivated by the fact that the clustering algorithm, such as Levin in Surratt, normally has higher accuracy than what we see from the visualization. For example, on this RNA mix dataset with the true labels, we know that the clustering algorithm will give us about 95% accuracy. While in the t-SNE and UMAP, different clusters may overlap and make us hard to tell the separation, which could be very confusing sometimes. So we aim to solve this problem by adopting the label information. Meanwhile, we also hope to alleviate two problems identified in the t-SNE and UMAP. The first is that they lose the large-scale geometry information, which will lead to the inaccurate cluster position. For example, this is a cancer cell line dataset. We looked into this data closely and found that cell lines from the same organs should be close to each other for this specific dataset. And we marked them in the similar colors. But from the visualization of t-SNE and UMAP, we could see some cell lines are placed incorrectly. For example, these lung and upper arrow digestive tract cancers, which are blue and green ones here. So they are not close to each other, which means that some part of the large-scale geometry information is lost during the visualization. And the second is that t-SNE and UMAP couldn't reflect the true cluster variance or cluster size. In this simulating dataset, we set the blue clusters to have the largest variance but the smallest sample size, and the green ones are opposite. And also the red clusters have the medium variance and the medium sample size. So from the visualization, we could see that the largest clusters are green ones due to the largest sample size instead of the true small variance. And we believe that these problems are partially caused by the crowding problem. And here is an example of the crowding problem. The data is a three-dimensional ball with two layers. Yellow points are at the outer layer and the blue ones are in the middle. When projected into two dimensions using MDS, these two non-overlapping groups will overlap. This is because the low dimensions don't have enough space to hold as many points as the high dimensions, while keeping the distance relationship. Where we say low dimensions only have a small capacity, which will cause the crowding problem. So in our method, we try to quantify the capacity so that we could solve the crowding problem more accurately. The capacity we define could be viewed as the number of points in the neighborhood associated with the radius, which is the circle on this manifold, and divided by the number of all the points on the entire manifold, given some distribution. Then we propose a new distance so that the capacity in the high and low dimensions we define could match after projection, and we call it capacity-adjusted distance. This changes any original distance defined by users in the high dimensions, such as Euclidean or geodesic distance, and could help to preserve the capacity so that the crowding problem is alleviated. Then we adopted the label information to improve the results of the original CPM, and we call it Supervised CPM. It mainly has three parts. First, we put different clusters a little bit far by multiplying a small constant larger than 1 on this capacity-adjusted distance. Next, we change both the high and low dimensional distances into two t distributions to preserve more geometry, and we optimize this objective function, which has two parts. The first is the KL divergence, and the second is a cluster separation measure. So by optimizing this objective function, we could get our final embedding Y, which has a larger separation between clusters. We also compared our method with UMAP, t-SNE, and supervised UMAP on several datasets. And the first is the synthetic dataset I used to show the cluster size preservation. So the SUP-CPM visualization reflects the true cluster size now, and you can see it from the last two figures. So it also recovers most of the isometric structure between different clusters. In this dataset, cluster with the similar color should form an equilateral triangle structure, and the distance between clusters with different colors should also be equal. So this figure shows the SuperCPM preserve most of this kind of geometry structure, while t-SNE and UMAP loses some part of it. And this one is the PBMC dataset. We could see that SuperCPM again separates different clusters, and the cluster positions are more resemble to the MDS results, especially for these platelet cells, which are pink ones here. So from MDS, we could see that platelet cells are very far away from others because platelet cells are very different from other immune cells. But t-SNE and UMAP don't reflect these features. You can see here the pink ones are very close to other cells, which is not correct. But SuperCPM placed these pink ones very far from others, which is correct. So we also have other visualization results and an objective metric comparison included in our article. But due to time limitations, I only present these two results. In summary, supervised capacity preserving mapping is a supervised visualization method that could alleviate the crowding problem using capacity-adjusted distance and label information. Besides that, it also has good performance in separating clusters, capturing cluster variance, and large-scale distances. As for the future direction, we are working with more flexible label information, such as continuous label or only a part of the more accurate labels. So our article has been published on the bioinformatics, and the code is also posted on the GitHub. You are very welcome to give it a try, and I'm looking forward to your response. At last, I want to thank Professor Yu Lei, Ron Wang, and Yun Xie for their advising and help, and also my current advisor, Professor Jessica Li, and my lab mates from GSB for their support and help. And also, thank you for your listening.